Okay, welcome everybody to this new seminar series, to this new seminar on speciation organized by the Network for the Integration of Speciation Research. This network is funded by the European Society of Evolutionary Biology, ESEB, and we've been organizing this seminar series since January 2022. We always alternate times to allow people from different time zones to join, and also all talks are recorded. The talk format is that we have two people giving a talk, about 1 hour, and then half an hour of general discussion, which is not recorded. We are also organizing workshops. We recently had one in Finland, and currently we are working on setting up a speciation database. There will be another speciation seminar in the beginning of April, and you can see here it's going to be at 5 p.m. Central European time. And there will also be a one-day symposium on perspectives on speciation that's organized by the Linnaean Society, with contributions from our network. It will be in person, where there's very few spots left, but you could still sign up, and also online, where there's unlimited spaces available. So if you're interested to join us on this one-day symposium, the link will be posted in the chat and you can sign up. As part of this, there will also be a special issue on speciation that's still accepting submissions until 31st of May. And that's going to be in the new Journal of Evolutionary Journal of the Linnaean Society. In this journal, we also recently published our paper that came out of the workshop of Finland toward the integration of speciation research. So if you're interested in that, please go and read it. But now I would like to welcome our speakers, Erin and Rachel. We're going to have a joint talk this time on paleontological views on speciation, entitled Reconciling Diversification Models in Paleoecology and Phylogenetics. I will start by introducing Erin and then hand over to Konrad to introduce Rachel. Erin Saupe is a paleobiologist investigating the interactions between life and environment over geological timescales. For instance, she studies how Earth and environmental processes affect biological diversification. She did her PhD at the University of Kansas in the US in paleontology, followed by a postdoc at Yale University with Derek Briggs. And since 2016, she is a professor of paleobiology at the University of Oxford in the UK. We think that it's very interesting to integrate a more paleo perspective on speciation, because we've had lots of talks focusing more on recent timescales, and Erin and Rachel focus more on a broader timescale. So we're very excited to hear what different perspective that brings to speciation research. I will now hand over to Konrad. Please go ahead.
Yeah, it's a great pleasure to introduce Rachel Warnock. Rachel Warnock is also a paleobiologist, and she works on computational tools and has made several really great contributions, in particular on dating and macroevolution. She's also been awarded the Hodgson Award. Actually, Erin also got that, so that's one thing that they share. Rachel did her PhD in Bristol with Philip Donoghue, and then did postdocs at the Smithsonian with Peter Wagner, and at the ETH in Zurich with Tanja Stadler. And she's currently a full professor in the geology department at the University of Erlangen. So Rachel or Erin, if you would like to share your slides, please. Super.
So hopefully everyone can see I'm doing it. I'll just hide. Yeah, so thank you very much for the invitation. I'm really honored to contribute a talk, and I'm really honored to be presenting alongside Erin today. So we're going to talk a lot about how we think about the process of speciation in different modeling contexts in paleobiology, paleoecology, and macroevolution. So here I'm showing plot of Phanerozoic diversity. So this was from the 1980s and was one of the first comprehensive attempts to quantify diversity over the past 540 million years. And it captures some of the major events in Earth's history that have affected biodiversity, including these major or mass extinction events, as well as intervals that are characterized by exponential increase in diversity. And this picture of diversity has changed as we've collected more data, and especially as we've developed new tools and new methods for handling incomplete sampling, which is extremely important to consider when working with the fossil record. And now we've moved towards asking many questions about the processes or the environmental controls, for example, on biodiversity. So we can ask questions about what promotes or inhibits diversification through time. So I'm going to talk mostly about phylogenetic models and to introduce a little bit about how we use phylogenetic trees in macroevolution. So we often think of trees as providing a framework for or a toolkit for allowing us to estimate evolutionary relationships. But if we can obtain a dated phylogenetic tree, then we gain a much richer understanding of a clade's history. So for example, we can place, if we can obtain a dated phylogeny, we can place the diversification and origination events in a tree within a geological context. And we can test hypotheses about what might have been going on in Earth's history that coincides with lineage diversification events. We can also, within a temporal framework, estimate rates. So we can calculate rates of molecular or morphological evolution. And we can also estimate diversification rates. So this includes species origination and extinction. So moving on to inference tools in macroevolution. So when we devise models to answer questions about or test hypotheses about what might have contributed to patterns of diversification and extinction through time, we typically think of three generating processes that are kind of at the core of many of the models that we use for inference or to estimate parameters in macroevolution and paleobiology. And this includes the speciation process or extinction process and fossil sampling process. And generally, to combine these processes, we use birth-death process models. And within these models, the speciation, extinction, and sampling process are stochastic, meaning that we have some rate associated with each one. And there's some probability or some rate with which these events occur, according to these stochastic models. And with these models, we can use them for both forward simulation, and we might do this for a number of reasons. For example, we might forward simulate data so that we can test models or test different methods, or we might use simulations to explore parameter space. Alternatively, we can use these models to perform inference. And this means we can apply these models to empirical data, empirical observations, and estimate macroevolutionary parameters that are of interest to us, assuming the processes that underlie these models. So if we want to take a step back and think about what's the most sort of simple set of assumptions we could make about a process that gave rise to diversity over time. So we might have through time, we have the generation of new lineages, so species origination events. And then we also have through time lineages that go extinct. And then we also have fossil sampling events. And the combination of these three processes is kind of the most simple set of assumptions that we could make about the processes that generated diversity through time. And these are at the core of the modeling framework that we use in this paper. So this structure here, if you forward simulate under a birth-death process model, is called the complete tree. But from observed data, we obtain the structure called the reconstructed tree. So this we obtain by pruning away all unsampled lineages. And using a model called the fossilized birth-death process, we can calculate the probability of observing a reconstructed tree with fossils along the internal branches. So before moving on to introducing the premise of our study, I wanted to just be a bit more explicit about what is assumed about the process of speciation by these models. So here are three different modes of speciation that we talk about in paleobiology. So we have, for example, this budding process, where essentially we have the continuation of the ancestral lineage. And we don't, so at the point of species origination, the ancestral lineage doesn't disappear. So this equates to one speciation event. In the middle here, we have a bifurcating speciation scenario. And here we have, at the point of divergence, we have the extinction or the disappearance of the ancestral lineage. And then we observe two new species under a bifurcating scenario. And so that's equivalent to two speciation events and one extinction event. And similarly, we can have anagenesis along internal lineages, where we have, so along a branch in a phylogenetic tree, you might see the evolution of one morphotaxon into another. And what's interesting is that although we draw phylogenetic trees as bifurcating structures, existing birth-death process models actually estimate the rate of budding speciation, because they basically every new divergence event leads to the addition of one new species in a phylogeny. But we observe differences between fossil-based estimates because, or one of the reasons we observe differences between fossil-based estimates of speciation and extinction might be that the fossil record reflects all three of these modes of speciation if we don't take into account the speciation process. And I've listed here some papers where we explore some of these issues. But that was just to make this very clear in the context of this study. So moving on to obtaining estimates of macroevolutionary parameters. So we can use a Bayesian phylogenetic inference approach. And if we combine the fossilized birth-death process with a substitution and clock model, we can obtain a dated phylogeny. So the core data that we use to infer dated trees, from which we can then go on and estimate rates of phenotypic evolution or diversification rates. The core data are phylogenetic character data, so morphology or molecular sequence data, and fossil age information. And then we have these three models that combine to allow us to calculate, to perform Bayesian inference under this tripartite approach. And so the substitution model and clock model allow us to model the evolution of phylogenetic characters along a tree. And then we can use the fossilized birth-death process or other birth-death sampling process models as the tree model. And this is where our fossil age information comes into it. And this is the framework that we use in this study here. So I will hand over to Erin shortly to talk about ecological niche models, but I already wanted to emphasize some of the differences. So within ecological niche models, the process of speciation is modeled completely differently. And essentially diversification is driven by climate and habitat. And here I have an example from Erin's work where this shows like a use case of ecological niche models in paleobiology, but we can look at the present day distribution of a given set of species, and then we can work out what their niche preferences are. And then we can project back in time and work out where those niches would have been in Earth's history in different parts of the globe. And then you can compare whether these predictions match up with the fossil record. And this is a really nice example where from birds, where we see that where we predict the niche space of different groups of birds to be is validated by the fossil record. In many senses, these ecological niche models capture the processes that we believe are sort of closer to how speciation actually occurs. So they're fundamentally very different to phylogenetic models and birth-death process models. And the question we wanted to ask is really, well, given that these models are potentially capturing some of the processes that we're most interested in testing hypotheses about, can we actually recover reliable phylogenetic parameter estimates using data simulated under ecological niche models? So our study questions were, can we reconstruct accurate phylogenies for data simulated under ecological niche models using the fossilized birth-death process? And then we also wanted to address how non-uniform or environmental dependent fossil sampling impacts the results. And before talking about the results, I'll hand over to Erin to discuss her modeling framework in a lot more detail. But just to give you an overview of our methods, so we simulated speciation extinction processes using ecological niche models, and these output phylogenies with known topology and divergence times. And then we simulated under different fossil sampling scenarios. And we simulated character data using standard phylogenetic models, and then performed inference using the fossilized birth-death process. And then because we have the true trees, which are output by the ecological niche models, we can then compare the inferred parameter estimates to those true values. So to walk you through some of the simulations, I'll just hand over to Erin.
So yeah, that was a really excellent introduction to our project by Rachel. And again, really thanks so much for inviting me and Rachel to speak. So as Rachel was saying, we are trying to combine two different models that think about diversification and speciation in deeper time. So my sort of eco-evolutionary niche model simulations and Rachel's FBD phylogenetic diversification models. So what I'll do now is briefly introduce the simulation framework that I use to examine diversity dynamics in deeper time, which are underpinning the simulations that we've been running with the FBD model that Rachel works with. And then I'll pass it over to Rachel again to talk about our results. So essentially, what I'm really interested in is thinking about how speciation and extinction occur over time in response to climate change. And the framework that I have developed is a virtual world where speciation and extinction are forced by climate change. So this framework is spatial in nature, and it allows virtual species to move about the landscape and to respond to climate change. Each simulation starts from a particular seed cell, which you can see here, and the species is afforded with a particular tolerance level to certain environmental or climatic conditions. And that dispersal ability that they're also afforded or assigned allows them to then disperse across the landscape and search for suitable conditions. As I said, this is a gridded world, which you can see here. And so what happens is that the environmental or climatic conditions in these grid cells change over time. And in this particular simulations that we're using here, the climatic changes are represented by a 120,000-year record of how we think climate changed from in the Pleistocene. So starting 120,000 years ago, and then stepping forward into the present day. And these environmental or climatic changes are cause species to have to move about the landscape, and certain pixels will become suitable or unsuitable for them depending on those tolerances. and the species have to react dynamically to these changing conditions. So in the modeling framework, as I mentioned, species can move across this gridded landscape based on the dispersal ability that they were assigned, based on their niche tolerances or niche breadth that they were assigned, and then they can only occupy areas that were climatically suitable for them. The environmental conditions of these cells, though, would change through time, and therefore it changes whether the actual grid cells remain suitable for the species. So in this way, the spatiotemporal dynamics of climate is affecting 3 different processes in these eco-evolutionary simulation models. So it's causing range shifts, it's causing speciation, and it's causing extinction. So in this simulation, speciation occurred through range fragmentation as a result of climate change. So if 2 populations of these virtual species were isolated by unsuitable climate, and this isolation persisted for a sufficient period of time, then speciation would result. Extinction would occur if all of the occupied suitable habitat for a species disappeared, or if a species couldn't keep pace with climate change based on their assigned dispersal ability, which this is sort of a graphic illustrating that. So thinking about speciation then in our model, speciation would occur allopatrically as a direct result of geographic isolation of 2 populations or 2 geographic ranges that were fragmented, and this fragmentation would occur because of the spatio-temporal dynamics of climate change. And that occurred because, of course, species have assigned tolerances and they have assigned dispersal abilities, so they can become isolated depending on that dispersal ability. Speciation would occur after the sufficient reproductive isolation, and in this way, the speciation concept in these eco-evolutionary simulation models is akin to the biological species concept. From each starting seed point or virtual species, the simulation records diversification dynamics, so the pattern of speciation and extinction over the time in the form of phylogenetic trees, as Rachel said. So each of those initial starting virtual seed points will result in some diversification process that's driven by the dynamics of climate change over time. So we have a phylogeny that gets recorded. And this simulation framework can be applied to generate all sorts of interesting sort of null expectations for diversity patterns and to interrogate mechanisms responsible for spatial diversity patterns. And in one of the first uses of this eco-evolutionary simulation model that we did, we were interested in studying the mechanisms that generate latitudinal diversity gradients. And of course, everyone knows latitudinal diversity gradient is one of the largest scale and longest known patterns in ecology, and first recognized over 200 years ago by Alexander von Humboldt, when he noted that diversity might be higher at the equator than at the poles. And ever since his discovery around 200 years ago, there have been numerous hypotheses proposed to explain why we see higher diversity in the tropics. And what's really exceptional, of course, about the diversity gradient is the pervasiveness of the pattern. So we find the diversity gradient on land, like you see here for vertebrate diversity. We find the diversity gradient in the sea, like you see here for copepod diversity. And then we find it not only in animals, but also in plants. And so, although numerous hypotheses have been proposed to explain the diversity gradient, it's really important to remember that the only processes that can actually generate differential patterns of biodiversity across space are speciation, extinction, or dispersal. And there's been a lot of debate about which of these processes contributes to the diversity gradient, or is it some combination of all of these? So speciation can be higher at low latitudes, which can contribute to a diversity gradient. Extinction can be higher at higher latitudes, which can contribute to a diversity gradient. And then dispersal, of course, can contribute to a diversity gradient if more species are entering the tropics than leaving the tropics. So that might also explain higher diversity. And so what we wanted to do was to actually try to explore which of these three processes might be contributing to the diversity gradient using this eco-evolutionary simulation framework. And then not only that, but try to understand and test a specific hypothesis, which is, can spatially structured heterogeneous climate change, so essentially changing climate across space and through time, can that explain higher diversity at low latitudes? And the hypothesis that we had was that the particular dynamics of climate change at low latitudes would fragment ranges more there, causing more geographic isolation. This geographic isolation would lead to enhanced allopatric speciation, and this would pile up species in the tropics, potentially creating or generating the latitudinal diversity gradient. So in other words, more fragmentation because of more spatially structured heterogeneous climate change, and this would result in speciation and higher rates of speciation at low latitudes, which would create the LDG. So using our eco-evolutionary simulation framework, we could actually attempt to replicate the latitudinal diversity gradient using only a few species characteristics, so that niche breadth and dispersal ability interacting with this dynamic climate. And as I mentioned, the dynamic climate that I used in this simulation study, and that is also used in the simulation work that I'm doing with Rachel that we're talking about, was a record of 120,000 year temperature and precipitation changes. So the framework is virtual and we use virtual species, but these virtual species were based on empirical data for vascular plants. So, and vascular plants, like many other groups, do exhibit a strong diversity gradient. So tropical forests, for example, are typically an order of magnitude more diverse than temperate forests. And the changing climate conditions, as I said, 120,000 year record over the Pleistocene. And these are realistic estimates of how we think climate changed over this interval of time, coming from coupled atmosphere ocean generalized circulation models. And these are specifically from Paul Valdez's group at the University of Bristol. So what you're seeing here is that year zero was 120,000 years ago, and then we're stepping forward into the present every 100 years. As I said, then each sort of initial simulation started from a particular point on Earth. So that's where the initial virtual species starts from. And they were afforded a particular niche breadth and dispersal ability, and then they could begin interacting with the environment, searching for suitable habitat. So suitable habitat for virtual species in this framework was defined very simply. So simply as rectangles, so within which all of the conditions were suitable. And we tested two different niche breadths, so a broad one represented by that large rectangle, and then a narrow one represented by the smaller rectangle. And we defined tolerances for both temperature as well as precipitation. So just to give you an idea of how this works, so the niche breadth dimensions for the virtual species in this simulation came from agriculturally important plant species. So there was a database of over 1,700 plant species from which we were able to take sort of realistic niche breadth definitions. And our narrow niche breadth species had a temperature tolerance of 12 degrees. And our broad one had a broader temperature tolerance. So I think it was 20 degrees. And so essentially, if you fell on a grid cell of 12 degrees Celsius, for example, so the initial virtual species started at 12 degrees, then you could afford a temperature tolerance of 4 to 20 degrees, for example. But if for the broad one, if you fell on 12 degrees, then you'd have a temperature tolerance of -8 to 32 degrees Celsius. And then similarly, if you started as a virtual species in, say, the tropics with a temperature tolerance of 25 degrees Celsius, then you would have a temperature tolerance of 17 to 33 for the narrow niche breadth, and broader for that broader niche breadth species. And same thing for precipitation as well. Species, as I said, could search for suitable habitat, so they disperse around the landscape based on a probabilistic stochastic function that's defined by exponential decay curves. So essentially, the steeper the decay curve, the less good the species is at dispersing long distances. And then we were able to test all combinations of dispersibility and niche breadth in these simulations. So we could hold a trait constant and examine the effect of that trait. So as I said, 3 different processes in this simulation: range shifts, speciation, and extinction. And the way that climate changes is heterogeneous across the surface of the Earth. And so this could prompt different macroevolutionary processes in different regions, so in low or high latitude regions. And from each starting seed point, we will generate this phylogenetic tree and this pattern of diversification, including patterns of speciation and extinction. So I'm just going to briefly talk about the fact that we could actually replicate empirical patterns fairly well in this ecoevolutionary simulation framework. So this is the latitudinal diversity gradient that emerged from our simulations, which looks remarkably similar to empirical diversity gradients. So this was giving us some confidence that we were able to simulate realistic diversification dynamics and potentially get at some underlying mechanism about what is causing these spatial patterns of diversity over time. And I think what's really important to remember is that we only we started from no diversity gradient. So we had one virtual species per, say, latitudinal band, and going from no diversity gradient or an even diversity gradient, we could create something that looks like this, which I think is pretty remarkable. And then we're able to actually look into what is causing that diversity gradient. So what's causing that spatial pattern of diversity across latitudes. And what becomes clear is that speciation at low latitudes is what's driving the generation of the diversity gradient in the ecoevolutionary simulations. So low latitude speciation is higher, and it's actually not extinction. So extinction, at least over the time frame of this ecoevolutionary simulation, was fairly constant across latitudes. And then we can ask, okay, well, if speciation is causing the latitudinal diversity gradient to emerge, and it's higher at low latitudes, what's causing this higher low latitude speciation? And what becomes clear is that it's the spatiotemporal dynamics of precipitation. So precipitation is actually causing more range fragmentation of these species at low latitudes. This is causing isolation of populations and then eventually driving allopatric speciation. And we can actually look at this in terms of sort of rates of speciation through time and what climate variable is actually driving that. And again, low latitude precipitation at low latitudes is what's driving the higher speciation there. So I think what's exciting is that the simulation framework is able to replicate empirical diversity patterns remarkably well. And this suggests that we might be able to sort of simulate how diversification plays out over time in a realistic way. And so these are the simulations that we thought, well, let's try to reconcile and understand how these spatial ecoevolutionary simulations can be reconstructed with FBD models. And so specifically, in the work that Rachel and I are working on together, we're focusing on simulations from my work that used only broad niche breadth and good dispersibility, as this resulted in trees that were much more manageable in size, so they didn't have as many species. The tree topologies resulting from each simulation served as the basis for these FBD analyses, as Rachel said. So we have this known truth for our phylogenetic trees resulting from all of these virtual species that had good dispersibilities and broad niche breadths. And then for each time interval of the simulations, I was able to provide distribution maps of where each species was found. So where grid cells were occupied by those lineages. And these distributional maps were then used to generate the fossil samples for the FBD model that Rachel talked about. And this was actually done by our friend and colleague, Simon Derrick. So he was able to actually sample these fossil lineages. And instead of using 1,200 time steps, which is what I had in my initial simulation study. So every 100 years, I had a simulation dynamic occurring. We focused only on 12 time steps, so we only sampled every 10,000 years instead of every 100 years. And again, this was just to keep things computationally feasible. We then simulated the sampling that Rachel mentioned. So we used 75 sample sites randomly from a vector of all possible grid cells in my simulation framework globally. And the reason that we chose 75 sites is that this approximates the number of well-dated fossil localities from a lot of Pleistocene stages. And of course, the Pleistocene is the time frame over which we are modeling these dynamics. It also actually maintained a feasible number of samples as well, which again kept computation time down. So if any of the randomly placed sample sites intersected with the geographic range of a species, then that species was recorded as found or not found. And that was based on a probabilistic function designed to mimic preservation potential in the fossil record, which I'll get to in a moment. Sampling locations that didn't fall, though, on a species' geographic range were discarded. And then we iterated this for each time step of the model. So we actually had new sets of sampling locations for each time slice, which mimics the shifting distribution of fossil localities through time and space. So it's trying to mimic the actual structure of the fossil record. So whether the occurrence or fossil was actually found was based on this probabilistic function, which I mentioned, determined by the climate of in that grid cell at that particular time. So the particular temperature and precipitation conditions of the grid cell actually determined whether we could find or recover that fossil sample. And we wanted to do this because, of course, the fossil record is filtered heavily. Taphonomic processes mean that we might not find occurrences because they don't get preserved in the fossil record. So this was our way of testing the influence of potential sort of taphonomy on our ability to reconstruct these trees. And it's generally thought that warmer and wetter conditions foster microbial growth. And this, of course, encourages degradation of biological materials, including vertebrate bones. And so you might not find fossils as easily in these warm and wet conditions, whereas cool and dry conditions exclude microbial growth and may allow fossil material to persist for longer. And so to test the impact of varying potential, so preservation potential in geographic space, we did two different designs. So one is we applied fossil sampling using this preservation model where preservation was geographically structured based on the climatic conditions in grid cells. And then we did one where it wasn't. It was geographically unstructured, and the preservation probability of these samples was constant across all of the grid cells. So yeah, with that, I will pass it back over to Rachel to talk about the results.
Oh, sorry. No problem. Okay, so hopefully you can see my screen again. So thanks very much, Erin, for describing the simulation framework so nicely. So here's an overview again of our our methodological pipeline. So we simulate the trees using these ecological niche models and then simulate incomplete fossil sampling. And then to keep things straightforward, we simulated phylogenetic character data. So we simulated a molecular alignment for all samples in the tree using standard phylogenetic models. And then we performed inference using the FBD process, and then we could compare our parameter estimates. So just to really emphasize, these are the key parameters that differ between our simulation and our inference. And hopefully, based on my description earlier and Erin's description, what you can see is that we are, this represents a massive violation of like the model assumptions that we're using for inference. So we performed inference using the fossilized birth-death process, which I described earlier. So for this, we used the software Beast2. And we assumed that diversification and sampling were constant through time. And we used the same model of character evolution for both simulation and inference. And this basically means that we can isolate the impact of violating the assumptions of the tree model, as opposed to the clock model or the substitution model. One of the challenges when going to take the data that was output from Erin and Simon's simulations was deciding how to handle what we might call in paleobiology abundance data. So when thinking about species at a given point in time in Erin's simulations, the same species can exist obviously multiple points in time in different grid cells. So the output of some of these simulations is that the same species is represented multiple times during the same discrete time interval. And this isn't a scenario that translates very readily into the assumptions that are made by the fossilized birth-death process, which essentially assumes that at a given instant in time, you can only have a lineage sampled once. So this forced us to make a decision about how to handle this data. And we basically decided that there were three possibilities. One was to pick one sample per species per interval, or to force all samples to be part of what we might call in paleobiology a stratigraphic range, so force all the samples to be ancestral to each other, or to just allow all samples to exist as as individual samples or sample tips within the tree. So we basically applied all three of these approaches. So just to summarize, we included like all samples with no constraints, all samples where we forced them to be a stratigraphic range. We forced them all to be sampled ancestors, and then we also picked one random sample per interval per species. And getting straight onto the results. So here I'm showing the results where we quantify the accuracy of the topology. So we know the true topology that's output from Erin's simulations, and then we can compare it to the estimates that we obtain using our Bayesian inference under the fossilized birth-death process. And here I'm showing the Robinson's fold distance, which is a measure of topological similarity. And basically, with this metric, lower is better. So if the Robinson's fold distance is 0, then the trees are identical. And along the bottom here, I'm showing like the results that we obtain using these different approaches to handling abundance data. And what we see here is that the results are fairly accurate. And I should also mention at this stage, I'm showing the results that we obtain when we assume that there is uniform spatial sampling. So these are the results where we didn't incorporate environment-dependent fossil sampling. So we see that the tree topology is relatively accurate, and this makes sense because the signal for the tree topology is largely driven by the molecular sequence data. So that's good that we can still recover the signal for this parameter. So now I'm showing the results for divergence times. And basically this is the coverage probability. So this is like the proportion of the node ages where the true value lies within the 95% credible intervals that we get out of our Bayesian inference. And in the case of this metric, having a higher coverage is better. Again, what we see is that the results are relatively accurate. We obtain the best results where we have one random sample per species per interval. And this could be because this is we believe the sampling strategy which best approximates the assumptions that are made by the fossilized birth death process. So we get lower accuracy for the divergence time estimates under these different approaches to handling abundance data. So then moving on to showing you the results where we have non-uniform spatial sampling. So this is Simon's model of environment-dependent fossil sampling. And here we see the results are not much different to those we obtained using the uniform spatial sampling. And what we see here is that we're still able to recover a reasonably reliable estimate of the topology. And maybe what I should also add is this is an average estimate of Robinson's fold across the entire posterior. So this, so just to clarify that. What's interesting here, so this is like the non-uniform spatial sampling that is for the divergence times. And we see quite a considerable drop here in the accuracy that we obtain under some abundance handling scenarios under the non-uniform spatial sampling. So what this shows us is that non-uniform spatial sampling actually diminishes our ability to recover accurate divergence times. So these are our results so far. So just to provide some conclusions before we move on to the discussion, what we found when we went about designing this simulation study is that reconciling the assumptions made by ecological niche models and birth-death process models that we use in macroevolution is actually not straightforward. So it took us quite a lot of effort to try and kind of come up with a simulation pipeline that made the most sense. And this is obviously because they're making very different assumptions about the speciation process. But we also show in this study that ecological niche models provide an alternative modeling framework for testing phylogenetic inference tools. And we hope that we can expand this approach in a meaningful way. Some of our results are very encouraging. Despite relatively extreme model violation, we can actually recover reasonable parameter estimates using the FBD model under some scenarios. In the study, we've only applied a fossilized birth death process that assumes that diversification rates are constant over time and across the tree. And so some of our future work is going to involve expanding this to ask whether time and lineage variable fossilized birth death process models can be used to both improve the accuracy of our parameter estimates, but also can we test hypotheses about the environmental drivers of species origination and extinction. So with that, I'd like to thank our amazing collaborators. So Joelle Burrido-Satani, who's ENS in Paris, is actually the project lead, and she's done a lot of work using simulations to explore the behavior of fossilized birth-death process models, including a recent paper that I cited earlier on how we handle taxonomy within the context of the FBD process. And there are a lot of important implications about the role of taxonomy in designing these studies. And as Erin mentioned, Simon Darroch, who's at Senckenberg in Frankfurt, played a big role in the simulation side of things, especially designing the environment-dependent fossil simulation scenario. So thank you very much, and I will, await your interesting questions. Oh, thank you.
So thanks both for this amazing talk. Anyone who has questions, please post them in the chat. So we have now time for some questions on the the actual seminar, and then there's a sort of more general discussion later on. And we'll keep the questions on the seminar sort of relatively short, maybe 10 minutes. So maybe can I start out with a question? I found this fascinating. We've been talking about integrating macro and micro evolution in this network for quite a while, and you've actually done it. And it's especially that spatially explicit model, in some ways, is a model of microevolution. And I'm wondering, I mean, this may be a question for Erin. Is it possible to? So, as I understood it, you set up a phylogenetic simulation, or you recorded phylogenetic data under this model. But is it possible to run a kind of more population genetic simulation that is spatially explicit? So have you thought about doing that, or do you think it matters? So this is the time scale over which you're running the simulation seems to be the time scale where the things that the micro people worry about, like incomplete lineage sorting and so on, really matter.
Yeah, that's a great question. So yes, we have thought about adding sort of more population genetic parameters to these simulations. And actually some existing ecoevolutionary simulations now are doing that more and more. And it's currently work that I have been discussing and delving into with my postdoc, actually, Tom Smith here at Oxford, is trying to reconcile these spatially explicit models that are forced by climate change on long timescales, but then inputting and underpinning it by actually more population genetic dynamics. But I haven't run any models like that yet. So it really, these ecoevolutionary simulation models that are underlying the work that Rachel and I have been doing with our collaborators, and that I showed in the results for the latitudinal diversity gradient, they're just based on this idea of speciation being driven by geographic isolation over time.
Great. There's a question here from Roger in the chat. Roger, do you want to ask your question?
Yeah, I could. I was just wondering whether once species have arisen by fragmentation, whether they can then overlap in distribution and therefore contribute to local diversity and are not excluded from one another by competition.
Yeah, that's a great question. So that's correct. So obviously, in order to for speciation to occur, they have to be geographically isolated, so they're not initially overlapping. But if climate changes such that that area becomes suitable, or they're able to traverse the barrier that was isolating them, then they can share the grid cells, so they can actually overlap in the future in the simulation. So in that sense, there is no competition or competitive exclusion. But again, once again, we are sort of my collaborator Hui Chao, who's an amazing modeler. He's been playing around with adding things like biotic interactions, trait evolution, including niche evolution, into these models. And so that does now exist. And also in ecoevolutionary other models, other ecoevolutionary simulation models like Genesis, it exists as well. So you can actually add those biotic interactions and ecological limits.
Yeah,
But not in the top.
Generally, it's amazing how much you can leave out and still get kind of reasonable answers.
Yeah, I think that was the thing. We obviously knew, we obviously knew that biotic interactions are probably important at some spatial scale, et cetera. But the reason that we actually kept our models so simple, at least initially, was that very reason. We wanted to basically say, okay, with really minimal assumptions, no biotic interactions, no ecological limits, only sort of assuming dispersal and assuming a niche breadth, and assuming really basic assumptions about how speciation works, can we generate sort of realistic patterns of biodiversity? And what does that then tell us about sort of the diversification process and what drives diversity gradients through time? So I think it's really quite telling, at least in my opinion, that even with such a short period of climate change, and with such minimal assumptions, we can generate something that looks remarkably similar to empirical diversity gradients.
So one related question, can you relax the assumption that the niche, as I understand it, currently the niche breadth is fixed and the species that you get out are all kind of ecologically equivalent? Is it possible to actually put evolution in? So, or
yes.
is this, yes.
I'm so glad you asked that, because so I just published a paper in Nature Ecology and Evolution with my colleague Huey Chow, and he's first author on that publication, and we did just that. So we tested a bunch of different niche evolution scenarios. So we tested adaptation to climate change, random walk, different rates, etc. So we had loads of different niche evolution trait scenarios. And we used the same general framework. And we saw, well, the main goal of that was to look at whether diversification rates, net diversification, was higher when you had more niche adaptation. And the answer was no, actually. So either it was equivalent or rates were higher when you had niche conservatism. And of course, that makes sense under the assumptions of the model where the climate change is driving range fragmentation and isolation. But it was an interesting sort of test. And we also then wanted to see, do patterns differ significantly when you allow for niche evolution? So both within lineages, but also across speciation events. And the answer was also no, which was really reassuring. So like certainly in some scenarios where we allowed loads of niche evolution, then you got wacky patterns and you couldn't generate a diversity gradient. But for a lot of the more reasonable, like trait evolution models, we still were able to replicate diversity gradients, and actually a bit better than we did in the original latitudinal diversity gradient study.
That's cool. Lots to think about. I mean, the allopatric thing, is that just a threshold that you're putting in? If the time is X, then it's two species.
Yeah, that's right. So it's just a time that they may remain isolated. And of course, like even if the climate is still unsuitable and still fragmenting the ranges, they could still, if they have a good enough dispersal ability or just, via stochastic processes, they might still disperse and exchange genes in a way, right? And so then that would reset the clock. So it really has to be sort of pure isolation, no dispersal between the two populations for a set period of time. And we tested a number of different times to speciation just to assess the sensitivity of the patterns to a time to speciation. But unfortunately, given the fact that the simulation is only 120,000 years long, you can't play with it too much, right? So we were constrained by the duration of the study. But there are, again, so now we're running these simulations with longer time periods, which is exciting. So we can play with longer times to speciation, etc. And then there are also now other ecoevolutionary simulation models, including Genesis, which again has more of that population genetic framework, which thinks about sort of genetic differentiation. So it's not just time to speciation, it's how much different genetic differences accrue through time. So there are other ways you could parameterize it, but in what I showed you, it's just a time.
Yeah. And there's a couple more questions in the chat from Marina. Marina, do you want to ask some of them?
Yes, I have many questions. Thanks for the very nice talk. So I guess my first question, and I failed to send this to everyone, apologies, is how the result that you got about the latitudinal diversity gradient, does it depend on the initial conditions? For example, where you put your species to start with?
Yeah, that's a great question. So not really. So what we did, we had lots and lots of different virtual species, basically in almost in every grid cell on Earth. The grid cells were 1 degree spatial resolution. And then what we did is bootstrap. So for each latitudinal band, we would just take one of those virtual species at random and then use those simulations to look at the resulting diversity patterns. So we could do that, many different times, 100 different times, and create 100 different diversity gradients and then look at the variation. So that's what I was showing you, why there were sort of like error bars on on the diversity gradient pattern that I showed you? And the answer is no. I mean, you do get variation, but it's not significant variation.
Maybe I should, or actually, I have another question, if you allow me, Conrad.
Of course.
Yeah, you applied here your model and framework to kind of think about and study speciation. But I guess you actually can maybe reduce the number of time steps, but increase the richness of the grid that you're looking at, kind of having more locations, and just look at how would the range of one single species change across time. And then have you tried to compare that to the existing species distribution models, or maybe to some existing data?
Yeah, that's a really good question. No, I haven't. But it would be really interesting to look at sort of changing geographic range dynamics over time for these virtual species, and then how well that matches sort of our knowledge about the fossil record for plants, for example, etc. So yeah, it would be cool to look at, but I haven't done that.
So there's one question in the chat here from Bert. I'm just going to read this out. So I have a question to wrap my head around the integration of the ecological niche modeling and the phylogenetics. Did the phylogenetic inference track diversification dynamics directly from the ecological simulations, or was independent data used to build the phylogenies?
Maybe I can try to answer this. So just to clarify, like the ecological niche models were only used to simulate the phylogenies. But the ecological niche models don't play a role in the inference. They only provide us with the trees that we then used to test the phylogenetic models. And then, sorry, Erin.
Also the fossil sample distributional data.
Yeah. Yeah. And then we simulate phylogenetic character data along those trees.
Yeah.
But
And
one possibility for the future is that, I mean, like one thing that is exciting to think about is like, how could we integrate more of what we understand about the speciation process from ecological niche models into a phylogenetic framework? And conceptually and theoretically, some of that is already possible, but we haven't tried that yet.
That's great. So the simulation of the character data is completely independent of the ecological niche simulation.
Well, it uses the trees.
Yes, of course, but the characters are not. You could sort of think about characters that have to do with the ecological niches or something like that in a model where maybe they do adapt a little bit, but that's not happening at the moment. I think that, yeah.
Yeah, that would be cool.
Cool. Maybe we should move on to the more general discussion. Let's